slide.
Okay, next slide.
Okay.
Okay, so I'll get back onto that. So anyway, I don't know what sort of background I'm talking to. I presume gynecologists and biologists in IVF. Who's not in IVF? Just to quickly go through it, we're looking at about 1/6 of the global population have some sort of fertility problems. Next slide, please. And that's about the same around the world. This is a very common graph that people use. So basically, 20% of couples have some sort of sterile problem. Next slide, please. Now, when we look in the normal fertilization reproductive cycle, obviously follicular development, hormones from the pituitary and the ovary, and the endometrium are all very highly synchronized, and that obviously is why we're a successful species and we get implantation at the right time. When we go into IVF, then of course we're messing things up a little because we're super ovulating the woman, and as we all know, super ovulation often leads to an asynchrony of the endometrium, and that is why we, and I think a lot of other groups around the world now, freeze all the blastocysts and transfer them in a subsequent cycle. The next slide, please. This is a typical scheme of what we're doing in IVF. So on day 1 of the period, day 3 of the period, we start daily injections of an follicle stimulating hormone in various formulations. Approximately 10 days of stimulation, then they get a trigger shot of something like Gonasi, and we retrieve the eggs 36 hours later. On day 1, they are inseminated by ICSI, so a sperm is injected into the egg, and development goes up to day 6. Now, today, most people would transfer blastocysts. In the old days, you would transfer on day 3. So now I would say that most people go into blastocysts on day 5, day 6. And as I said before, the tendency today is to freeze on day 5, day 6, because the uterus is probably going out of synchrony. Next slide, please. So if we look at a typical development of an embryo in the human, this is a 2-cell, 4-cell division. Bottom right is the time in hours. We're looking in time lapse, of course. As we lose the formation of this cellular structure, that's the morula. And then obviously at the end, we get the formation of the blastocele and the blastocyst. So the blastocyst, this is a hatching blastocyst, but the blastocyst at this stage is then frozen, vitrified. So that gives us a time point or a fixed part of the cycle. So all the patients are receiving blastocyst frozen at that stage. Half of the patients approximately were homologous patients, so they're receiving their own eggs fertilized by the husband's sperm, and the other half were heterologous, so they're donated oocytes fertilized by the husband's sperm. Now, the sperm we did not select in any way the patients. They were randomly distributed to the control group or the treated group.
Sorry, Professor, you muted yourself.
Okay, can you hear me?
Yep.
Okay, sorry about that. So the only exclusion feature was in women with problems, pathological problems of the cervix. They were excluded. Next slide, please. Can you get the next slide? Okay, great. So in IVF, so we have frozen embryos going into the uterus. The uterus is obviously a black box. There's not a lot known about it. There's not been an awful lot of research on it. It's not easy to research on it. And in IVF, there's lots of treatments that people have used called add-ons, which can be from genetic chromosomal analysis of the embryo to the scratching of the endometrial surface to it to try to improve implantation. Now, all these add-ons are very controversial, and they are based on the individual patients and what we think, what we would think they needed or what would help in their particular situation. In this interesting study with carers flow, we took a totally different view of the whole procedure in a way, and we decided to irrigate oxygen into the vagina, which was previously known by the people at Carers Flow to be very useful for several pathologies in ladies, particularly in menopause or perimenopause, and could change the microbiome in the vagina to curing pathologies and pain and dryness in the vagina and lichens and all sorts of things that it's used for. So our question was, if we could change the physiology of the uterus by using oxygen, and obviously it's quite non-invasive, it's actually very pleasant to the patient, and so they receive, next slide please. So this is going a bit back to the frozen embryo, advantages of the frozen embryo. So the advantage of having frozen embryos is we're not restricted so much to the window of implantation is easier to calculate and to monitor, but just by ultrasound or whatever we're doing. And of course, if the endometrium isn't developing well, we don't need to thaw the embryo, and we'll do it the month later to avoid that we do not ruin a blastocyst. Next slide, please. So getting on to Carers Flow, it's been developed by the team at Carers Flow with lots of doctors in Italy. Basically using two very simple but very important components: high oxygen concentration and hyaluronic acid. And both of these elements individually are known to be beneficial for various reasons in the vagina. So we are at the vagina at the moment. Next slide, please. The oxygen in particular can be involved in stimulating growth factors. It could be involved in angiogenesis. All these pathways that I've put onto the slide, these are known to be induced by oxygen in all sorts of cellular tissue. Next slide, please. So these are the known uses at the moment of Carers Flow: vaginal atrophy, candidiasis, incontinence, lichens, burning, dryness of the vagina. Next slide, please. So what we have done is used Carers Flow as a stimulation of the physiology of the uterus. And what we did is give these 3 to 5 50-minute Carers Flow sessions. Next slide, please. And the flow rate is about 2 liters a minute, containing 0.2 hyaluronic acid. The pressure is 1 bar. So this is a very interesting machine that can be used in outpatient situations, which actually takes air from the surrounding and concentrated into high purity oxygen at about 93–95%. Next slide, please. So in the first sessions, we did mainly 3 15-minute sessions. The first session was 14 days before transfer. The second session was 7 days before transfer. The last session was about 24 hours before transfer. And in certain cases, when we could manage it, because obviously these patients have to come in to have this treatment, up to 5 equidistant sessions before the transfer. We did altogether in the first trial that's been sent off for publication, 377 patients. The treated were 161. Next slide, please. And one thing we observed straight away is that we could get a more healthy looking cervix just by this treatment. Next slide, please. So these are the results. So overall, I'll show the graph is the next slide, but overall we found a statistically significant increase in pregnancy, in implantation rate using Kereslow. When we break down homologous patients and heterologous obviously are the donor patients, it was highly statistically significant in the heterologous patients. The next slide will show you the table. So those are the actual numbers, and you can see in the heterologa, which is the egg donation patients, a very large increase over our baseline. Our baseline sort of implantation rates around 44%, 43%. And in all cases, an increase, but highly significantly in the heterologous population. Now, of course, the embryos are the same. We have 4 arms to this study. So, you know, we know the embryos are equal in each study. The only difference with the heterologous population is the age of the patient. So the typical age of a donor recipient patient is about 46 years of age, whereas an homologous patient is 38 on average. So that is obviously giving us some sort of signal that the uterus of a woman in perimenopause or menopause can be tweaked a little bit with oxygen to make it more receptive. Next slide, please. We don't know what's happening. This is the beginning of an exciting study, and we're asking colleagues around the world basically if they would like to get involved in this project. What we're doing in Naples is, as far as we can do, is we're interested in 2 things in the uterus that appear to us obvious targets. The first is the microbiome in the uterus. There's a lot of work on the microbiome in the vagina, very little on the uterus. So maybe the oxygen, the Kereslow treatment is improving the microbiome in the uterus. And if it does that, parallel with that, or one of the reasons that we could change the physiology of the uterus is we could change the pH in the uterus. Now, for an embryo to implant into the uterus, it has to produce enzymes. We know that proteolytic enzymes. Any enzyme is the activity of an enzyme depends on the pH. The contractility of the uterus, which is another problem, as we all know, doing transfers, is also pH dependent. So pH is a very important factor in the uterus, and then certainly in the implantation process. Now, we studied years ago, about 30 years ago now, I think it was, the capacity of human embryos to regulate pH. And the blastocyst has a very good capacity to regulate acid or alkaline conditions. In the animal world, there are variations in pH during the estrus cycle. We don't know anything about in the human cycle. So we have we're investigating it at the moment. The microbiome is a little easier because it just means we can take a swab from the uterus before and after treatment and see if we're changing the flora in the uterus. Implantation, I don't need to say, is probably the most difficult thing and the most complex thing in the human body. So you have you have these different tissues that have to interact and they change in time. So first is the apposition of the blastocyst. Then there's a very strong adhesion, and then there's the invasion, which eventually leads to the formation of new blood vessels to help the blastocyst invade. There's so many growth factors involved. Years ago, people invented the error test to try and determine when the right time was to put a blastocyst into a uterus. But I think generally most people now agree that the error test is not very reliable in this moment. Next slide, please. Okay, so this is the last slide. This is where we are. Blastocyst implantation is extremely complex. We've always known that it's necessary to have a good oxygen supply in the uterus and nutrients in the uterus. And maybe we can modify this by applying oxygen and hyaluronic acid to the vagina. And as I say, it's non-invasive, so it's very easy to apply to the lady by a machine that's very useful and easy to use. Oxygen, of course, we could be looking at all sorts of redox reactions, reducing inflammation, growth factors, angiogenesis, so many things. And that's why we would like to open discussion to all our colleagues to see if people will start studying this effect. We could even be looking at a very simpler effect. Now, I've always worked with many clinicians, and I'm sure you know that some clinicians apparently are better at doing embryo transfers than others. And so it could be a simple effect of improving the physiology of the cervical canal and allowing us to recruit patients that otherwise would fail because they could be a bad transfer, or the uterus is so provocated by the transfer it actually contracts and the embryos get pushed out of the uterus straight away. We could, and we're thinking of this at the moment, we could be widening the implantation window, which is normally 24 hours, and that also would account for having more pregnancies. So this is the state of the art at the moment. We're continuing our clinical trial. We would like to work with as many people as we can to improve the numbers and to prove, obviously, if it's correct, and if it's correct, what exactly is happening in the uterus. Okay, I finished. Thank you, Nevis.